Very good afternoon to everyone. Welcome in the class. Let's crack CSIR UGC NET together. That is the name of our channel. Let's crack Gate 2021 together. You're watching this video on the India's largest learning platform, that is Unacademy. I'm your educator Farida Johar Bandukwala. I have a teaching experience of 10 years, qualified MP SLET examination in year 2018. If you're opting for Unacademy, what all things you are going to get? You are going to get daily live classes, daily, so that you can be always in that momentum of studies. Live means you can ask your doubt directly with the faculty members while the class is going on. The syllabus is very much structured. That means the whole thing you can complete in a small span of time. As for whatever time of competitive examination it is, they try to keep so many things in the syllabus so you can complete it with the help of an academy. And live tests and quizzes are taken so that you can evaluate yourself that how much you have learned so far, and unlimited access towards all the videos and towards all the preparatory material which has been given in that particular category. So Unacademy gives free live classes on YouTube, and it gives free live classes on a special platform. So the YouTube classes are completely free and special classes are also completely free. For YouTube class, you just need to go to the channel, subscribe to the channel, and you need to just sit and watch the videos. And for a special class, that is also very simple. You just need to download the Unacademy Learning app from the Google Play Store. After downloading the app, what you need to do is you need to make your account. After you have made your account, you need to use the referral code of an educator for attending the special live classes completely free. You can use my referral code, that is FB01. So do not forget to use my referral code FB01 so that you can be attending the special classes of mine as well as other educators. After that, there is this plus subscription. Plus subscription is a paid subscription, but do not worry because the amount is very much pocket friendly. For 12 months, it is all only 11,550 rupees. For 24 months, it is 16,800 rupees. And you can get an additional 10% discount if you're using my referral code, that is FB01. By using my referral code, you will be getting a 10% discount. So the 12 monthly syllabus is going to cost you 10,395 and 24 monthly is going to cost you 15,120 rupees. So coming to the topic, that is northern and western blotting. In yesterday's class we have done southern blotting. Today we are going to talk about northern blotting. So before beginning with the northern blotting, what I would suggest you people is that go and watch the video of southern blotting so that the basic things you will come to know. So starting with the proper topic, northern blotting. Northern blotting is a technique for detection of a specific RNA sequence. It is for detection of specific RNA sequences. This technique, northern blotting, was actually developed by James Alvin and George Stark at Stanford University, and it was named by analogy. Like southern blotting was already there for DNA, so the next technique, which is just a version of southern blotting, the name given was northern blotting. So it was developed by, this technique was developed by James Alvin and George Stark. James Alvin and George Stark. Right. Now, what are the steps which are involved in northern blotting? We'll be directly coming to the steps. The first step in the northern blotting technique is RNA isolation. RNA isolation. RNA is isolated from various biological samples, various tissues, various developmental stages. So RNA is isolated, and RNA is going to differ for different stages, for different developmental stages, for different tissues. So RNA is more susceptible to DNA to degradation as compared to DNA. That must be always kept in mind. RNA is more fragile than DNA. Always keep this thing in mind. After that, the samples, they are loaded on gel, and RNA samples are separated according to their size on the agarose gel. Gel electrophoresis for separation of RNA on agarose. So after the gel is run, the gel obtained will be containing different bands of RNA. Now this gel is blotted onto a nylon membrane or a nitrocellulose filter paper by creating a sandwich-like arrangement which we have seen in detail in yesterday's class. So that sandwich thing, you can just go and watch the video of southern blotting. So we'll be writing over here, gel is then sandwiched in filter paper to get a blot. So the paper which is used in this case can be nylon membrane or it can be nitrocellulose filter paper. After this, the membrane is placed in a dish containing hybridization buffer with labeled probe. So the membrane which we have got will be kept in the hybridization buffer which are containing the probes. Probes may be labeled radioactively or non-radioactively. So the probe, they are going to hybridize with the RNA on the blot and they are going to correspond to the sequence of interest, right? Whichever interest we want to identify, they are going to bind to that interest. So membrane is kept in hybridization buffer with labeled probe with labeled probe, and RNA is going to bind. RNA will bind to the specific sequence. Then the membrane would be washed out so that all the unbounded probes are removed. Washing will be done for removing all the things which are going to create unnecessary uh, disturbance in the actual result. So washing will be performed. After washing, the labeled probes are detected via autoradiography or via chemiluminescence, whatever kind of label we are going to use. So in both the cases, this results in the formation of a dark bands on the X-ray film. So labeled probes are detected via labeled probe detected via autoradiography or chemiluminescence, and we are going to get the result in the form of dark bands on X-ray film. Result in form of dark bands on X-ray film. Now the expression pattern of the sequence of interest in the different samples can be compared and can be analyzed. So this is the technique of performing the northern blotting. Now if we are talking about northern blotting, we must know that what is the application of this technique. So what are the applications of northern blotting? A standard for the study of gene expression at the level of mRNA can be done through northern blotting. Study of gene expression at mRNA level. Then detection of mRNA transcripts. Detection of mRNA transcripts. Then study of RNA degradation, that how it is degraded. Studying RNA degradation. Then to study RNA splicing. To study the half-life of RNA, and it is also often used to confirm the knockout mice and check the transgenic animals. Confirm and check transgenic oblique knockout mice, oblique knockout mice or animals. These are the application. Now we are saying application, and whenever we talk about application, it is kind of the uses, and it should also be considered that what all are the disadvantages of the technique. Without disadvantages, it is like we need to know so that further corrections and refining can be done. So the standard northern blot method is a relatively less sensitive than the nuclease protection assays and RT-PCR. Always keep in mind, less sensitive, less sensitive than nuclease protection assays and RT-PCR. Then detection with multiple probes is a problem. You cannot go for multiple sequence identification in the same process. So multiple probes can be a problem. Detection with multiple probes is a problem. After that, if RNA samples are even slightly degraded by RNAs, the quality of DNA and quantitation of expression is quite negatively affected. So degradation is a thing which we cannot afford at all in northern blotting. So slight degradation by RNAs is going to give a very poor quality data. Slightest degradation gives poor data. So this is our northern blotting. We have already done southern blotting, so we have started the thing from the basic, not from the basic, but from the advanced stage. So go back and watch the southern blotting, and now the next is western blotting. Right, the next thing is western blotting. So let's start with western blotting. Western blotting. So this western blotting is also called as protein blotting. Protein blotting. It is done for protein. As southern blotting is for DNA, northern blotting is for RNA, western blotting is for protein. So protein blotting is an analytical method and it involves the immobilization of protein on the membrane before detection using monoclonal or polyclonal antibodies. There we were using probes. Here we are going to use the monoclonal or polyclonal antibodies. There are different blotting protocols such as dot blot, 2D blot, etc. One of them most powerful is Western blotting, right? So protein blotting is a word which is given for Western blotting, and protein blotting itself can be composed of Western blotting or dot blot or 2D blot. So Western blotting we are going to see. Now we are going to see the principle of Western blotting. Principle of the technique. So Western blotting is an immunoblotting technique which will rely on the specificity of binding between a molecule of interest and a probe to allow the detection of molecule of interest in the mixture of many other similar proteins or molecules. I hope you have understood what I was trying to say. So it's an immunological technique. We'll write down the points. It's a immunoblotting technique. The immunoblotting technique. The immunoblotting technique, which rely on a specificity of binding between a molecule of interest and a probe to allow the detection of the molecule, right? So in Western blotting, the molecule of interest is a protein and probe is typically an antibody which is raised against that particular protein. So it's an immunoblotting technique. Molecule of interest, molecule of interest is protein, and in this case the probe is typically antibody which is raised against particular protein, against particular protein. So the SDS page technique is a prerequisite for Western blotting. We must perform SDS page, and the whole thing should be available for going for Western blotting. So SDS page is a prerequisite. Without it, it cannot go further. So Western blotting, which is also called as protein blotting, or it is also called as immunoblotting. Western blotting is also called as immunoblotting. Immunoblotting. So it is a technique for detecting the specific proteins which are separated by electrophoresis by use of labeled antibodies. So called, so it is called as immunoblotting, and it is quite similar to southern blotting. So that's why the name is given in analogy, southern blotting, western blotting, right? So this is the thing. Now, Western blot is an analytical technique, and it is used to detect the specific protein from a mixture of samples derived from a tissue homogenate or extract. From so many things, we have to just identify a single kind of protein. Now, before moving further, before moving with the steps, as we have seen the advantages and disadvantages in all of the cases, we'll be starting Western blotting with the advantages. That what are the advantages of Western blotting? So, the analysis by Western blot can analyze any protein sample from cells or tissues, but it also can analyze the recombinant proteins which are synthesized in vitro. So any protein, recombinant protein, Western blot can be used. Then Western blot is dependent on the quality of antibody which is used to probe for your protein of interest and how specific it is. So it is completely dependent that how specific antibody we have. Then we said that it cannot go without SDS page. So the gel electrophoresis, so use of gel electrophoresis to separate the native and denatured protein by the length of polypeptide. So that is how the separation would occur, and then they would be transferred on the nitrocellulose paper or nylon paper or polyvinyl papers. They'll be taken on them, and then they'll be baked or they'll be heated at such a temperature that the bands are fixed, the protein bands are fixed, and then they'll be flooded with antibodies. And antibodies, they are going to bind at certain places where they are getting the protein to which, for which it is formed. Then there can be some kind of colored reaction which is going to give us the result. So monoclonal antibodies and polyclonal antibodies are used. Mostly monoclonal antibodies serve as the best thing when we are performing Western blotting. So this method is used in the field of molecular biology, biochemistry, immunogenetics, and molecular disciplines of various other types, genetic engineering and whatnot. Now we have seen the basics and everything. Now coming to the actual step, that how it is performed. So it is basically composed of 6 steps. First one is tissue preparation, then gene electrophoresis, then transfer, then blocking, then detection, and then analysis. We are going to talk about the steps in detail. First one is tissue preparation. Tissue preparation. So samples can be taken from the whole tissue or from the cell culture, and in most of the cases solid tissues are first needed to be broken down, and then the cell extract is used. And another thing that should be kept in mind while the tissue preparation is there shall be no contamination of microorganism. There shall be no bacteria, no virus, or any kind of environmental sample which is making it impure. So Western blotting is basically not restricted to the cellular studies only, so there should be no kind of contaminated contamination. It should be noted that bacteria, virus, or environmental samples should not contaminate the whole thing. And bacteria, viruses, and environmental sources, they can also serve as the sample. Not only the tissue lysates, but bacteria, they can also be used for further detection of a certain kind of antigen in them, for which we have developed an antibody. Those all things can be done. So assorted detergents, salts, buffers, they can be employed to encourage the lysis of cells and to solubilize the proteins. Tissue preparation is often done at very cold temperature to avoid the protein denaturation. This is the thing. So tissue preparation, we will write that whole tissue can be taken, whole tissue or cells from culture, cells from culture or bacteria or viruses or fungi can be used, but it should be free from contaminants. Free from contaminants. So various different type of detergents and salts, and they can be employed to solubilize the protein, right? So sorry. So another problem which can arise over here is of denaturation of protein. So to avoid this problem of protein denaturation, the whole process is done at cold temperature. Cold temperature is employed for this whole thing. After that, we are going to see gel electrophoresis when we have to separate out the protein. So we will be doing the gel electrophoresis. So the protein of samples, they are separated by using the gel electrophoresis. Separation of protein can be done by isoelectric point, molecular weight, electric charge, or combination. So 2D gel electrophoresis, 2D electrophoresis, or isoelectric focusing, any kind of gel electrophoretic technique can be employed to separate. So the principle involved is the difference in electrophoretic mobility of different kind of proteins. So SDA's page sample and SDA's page would be the most preferred separating technique. So SDA's page most most preferred. Right. After that, after gel electrophoresis, we are going to see what is this transferring. Transferring. So in order to make the proteins accessible to the antibody, we cannot put it anywhere. We need to make it accessible to the antibody detection. They are moved from the gel onto the membrane, which can be made up of nitrocellulose, polyvinyl, or any other thing. So the membrane is placed on the top of the gel in a very sandwich manner, and they are stacked with the filter papers on the top, and the whole thing can be dipped in the buffer, and from above there will be weight which will be kept, so that due to the capillary action, the whole thing is going to move to the membrane. The whole thing is going to stick to the membrane. This is called as transferring. Transferring. So transfer of protein bands on nitrocellulose filter paper, or we can see polyvinylidene difluoride paper. This paper can also be used. So another method which is used for transferring, the simple blotting, we have seen simple blotting technique of transfer. That's it is also called as Western blotting. The blotting word is not left out because this whole transfer is called as blotting. So another method for transferring the protein is called as electro blotting. That was simple blotting. This is another thing is electro blotting. Electro blotting. So in electro blotting, there will be use of electric current to pull the proteins from the gel into the nitrocellulose filter paper or polyvinylidene difluoride paper. The whole thing will be actually transferred with the help of the electric current. That is called as blotting. So next is blocking. So that's why the word blocking came. So next is blocking. Now the membrane has the ability to bind the protein in both the target and the antibody. Target and the antibodies are protein. So there should be there would be unwanted attachment of antibodies to the paper, and that we don't want. So blocking of non-specific binding is achieved by placing the whole membrane. Now for example, this is the membrane and it is having some kind of bands like this over here, and rest of the place is completely, this whole place is like completely free for binding with anything. So this place can be always available for binding with the antibody. We don't want this to happen. So what we are going to do, we are going to block the sides. And how are we going to block the site? By placing the membrane in a dilute solution of protein, typically bovine serum albumin. So how we'll be doing? We're taking the highlighter, and now these whole places where the unwanted binding of antibody could occur, we have just covered it with the BSA. So like this, the whole thing is now blocked. Now no unwanted binding can happen. So this thing is your bovine serum albumin with a minute percentage of detergent, that is 20. So this is blocking. So the protein in the dilute solution attaches to the membrane in all the places where the target protein have not attached, and when antibody is added, there is no room on the membrane for the antibody to attach at any other place. So this is how the blocking is performed. Very important, otherwise we'll get the wrong results. After blocking, the next step is detection. Detection. So during the detection process, the membrane is probed for the protein of interest with a modified antibody, which is linked to the reporter enzyme, which when exposed to appropriate substrate is going to give a color, and which can further be subjected to the colorimetric reactions. So during detection, what we'll do, the membrane will be probed. Membrane is probed for the protein of interest, for the protein of interest with modified antibody, with modified antibody. And this modified antibody is actually linked with a reporter enzyme. And when this whole thing is exposed, this whole thing is exposed to substrate, this is going to give a colored reaction. Colored reaction. And this is how the detection can occur in Western blotting. After detection, the next step is analysis. Analysis. So after the unbound probes are washed away, the Western blot is ready for detection of the probes that are labeled and bound to the protein of interest. So size approximations are taken by comparing the stained bands to that of the marker loaded during the electrophoresis. So the process is repeated for the structural proteins such as actin or tubulin that should not change between the samples. So probes would be washed, and then the whole thing will be analyzed for any kind of colored reaction. So that was the analysis. Now, whenever we are talking about Western blot, what are the advantages? Because advantages and disadvantages and all those things are very important. So, talking about ELISA is a non-specific test. Western blotting is more specific test as compared to ELISA for detection of HIV. So, more sensitive than ELISA for HIV. Then it can detect one protein in a mixture of proteins while giving information about the size of the protein and about the specificity. So can detect one protein in a thousands of proteins, can detect one protein from thousands of proteins, from thousands of proteins. So Western blot is also referred to as gold standard, the gold standard technique. It also tells you how much protein has accumulated in the cell. So along with the qualitative analysis, it also serves at many a times as a quantitative analysis. Western blot is used a lot in clinical medicine as well. Clinical medicine also, it is used highly. So the confirmatory HIV test employs a Western blot to detect the anti-HIV antibody in the human serum sample. So that is the thing. Then Western blot is also used as the definitive test for bovine spongiform encephalopathy (BSE), which is also called as mad cow disease. So clinical method for HIV, for mad cow disease, and in some form of the Lyme disease also Western blot is employed. Then Western blot, we have seen that it is a confirmatory test for HIV, because if antibody is there, then it's a confirmatory test. So virus is enveloped with a different type of proteins. So the detection of these proteins are useful in detection of the virus. So Western blot helps in detection of the proteins which are present in the covering of the HIV. Now, what are the disadvantages of this technique? Disadvantages: the protein, they are quickly degraded, so it has to be done very carefully. So Western blotting won't detect it well if they are degraded. Protein degrade fast, protein degrade fast. Then this test takes longer than the other existing test, so longer, so that's why it is not preferred. Then it can be costly as well, because various techniques are involved and monoclonal antibody, they need to be generated. So it is itself a quite difficult thing to be performed, because it won't be cheaper technique, right? So like this, we have completed this topic of northern and western blotting. So thank you very much. And as we say in the starting and in the end, let's crack CSIR UGC NET and GATE 2021 together. That is the name of our channel as well. Let's crack CSIR UGC NET. Hit the bell icon for getting all the notifications of the upcoming video. And if you haven't subscribed the channel, subscribe our channel, and it would be really appreciated if you like my video. And for getting the plus subscription, you can use my referral code FB01. Recently, a discount is going on in the plus subscription. So do not forget to use uh, this opportunity of getting a great discount. And again, a 10% discount can be obtained by using the referral code FB01. And then the special classes which are completely free, for them also you will need my referral code FB01. You can watch my special class as well as the special classes of other educators by using my referral code FB01. And that is it for the class. Thank you very much for your patient listening. If you really like the video, give it a thumbs up and write in the chat box. And if you have any kind of doubts and queries, I'll be really happy to take up your doubts and queries. Just write down in the chat box and I'll take up your doubts and queries, and if you want me to make a certain video on a topic of demand, I'll be really glad to make a video on that too. So this is it for now. Hope to see you again in another class of mine. It's a bye from my side.